Our next topic is types of animal cell cultures. Now, there are two main types, which includes organ culture and explant culture. In explant culture, the tissue is removed, it is chopped finely with the help of scalpel or scissors or any other technique, and then number of cells are calculated and inoculated into the medium. So here tissue is at solid liquid interface and cell migrates to form outgrowth. And in organ culture, tissue is at gas liquid interface. Histological structure here in this organ culture is maintained as it is. See here, organ is removed, say for example, it is chopped and then the cells are inoculated, and in organ culture, organ is removed, placed on a cover slip, and then that cover slip is put in a container or say plate containing liquid medium. Okay, so these are main types of tissue culture. Then these are advanced methods which is known as dissociated cell culture and organotypic cell culture. See here, you can see in this organotypic cell culture, a gel matrix is present or fibers are present which supports the cell growth. Cell adhere to the matrix and then grow. And here the cells are suspended. In dissociated cell culture, the cells are suspended. See, this is like a fermenter. It is continuously stirred, and then cells, due to this stirring, cells get dissociated from the piece of tissue. Single cells are released and disaggregated tissues are then grown in a liquid medium. Okay. See, in dissociated culture, disaggregated tissues, cells from monolayer at solid liquid interface, and here different cells co-cultured with or without matrix. Organotypic structure is created. Organotypic structure means the same condition as the cells are grown in vivo, that is inside of the body, is created artificially so that cells can feel the environment. These are properties of different cell types. This is the category organ culture, explant and cell culture. Sources for each type of cell culture may be different. Like for organ culture, embryos can be taken or adult tissues can be taken. For explant culture, any tissue fragment can be grown. And for cell culture, disaggregated tissues or primary culture or propagated cell lines. Organ cultures require high efforts because you have to maintain organ as it is in vitro conditions. For explant culture, moderate efforts are required, and for cell culture, low efforts are required since the culture is already established. You just have to grow the cells in vitro. So you don't get confused between in vivo and in vitro. In vivo means inside the body of organism or in natural environment, and in vitro is inside the glass or say in artificial environment in laboratories. Okay, so these are the other properties like characterization, histology, biochemical differentiation, propagation, sampling, reproducibility and quantitation. Subculturing advantages and disadvantages. This we'll discuss later on because this is another chapter included in your syllabus. We'll just have overlook advantages is cell can be propagated. You get more cells when you subculture. Possibility of cloning is increased. Increased homogeneity, that is, you can grow one cell type in one, say, flask. Homogeneous culture. Heterogeneity is different cell types grown in a one flask. Homogeneity, single cell type grown in vitro. Okay. Characterization of replicate samples and frozen storage. Disadvantages: trauma of enzymatic or mechanical disaggregation. Selection of cells adapted to the culture is difficult. Overgrowth of unspecialized cells or stromal cells. Unspecialized cells means cancer cells which do not have any specific character. They just grow and grow and grow. Genetically unstable. After subculture, cell may lose their genetic content. Cells may get unstable because of changes in ploidy level, loss of differentiated properties, and increased risk of misidentification or cross-contamination. As you subculture, there are chances of cross-contamination. Cross-contamination is contamination of one cell type with the other cell type. If you are working on two cell types together under one laminar hood, there are chances that, say you are, one minute, say if you are working on cell number 1 and cell type number 2. Say first you have subcultured this cell number 1. Say this is particularly HeLa cell line. Okay. You have subcultured this HeLa cell line. You have inoculated this HeLa cell line into the flask. Then, after completing this procedure of subculturing, second cell line you are taking, which is CHO cell line, Chinese hamster ovary cell line, and putting it in another flask. Now these two cell lines are different, but while working on this you are using same pipettes, same medium, so there are chances that these HeLa cell lines get mixed with the CHO cell line. So this is known as cross contamination. These are basic steps of cell culture. First, you have to choose a healthy individual. Then you have to choose explant, whether you are going to use embryo or whether you are going to use any other organ, that you have to choose. Whether you are going to choose eggs. Okay. Then organ is extracted. Okay. This tissue is then finely chopped either with a scalpel or enzymatically disaggregated. There are certain enzymes used in animal cell culture like trypsin. Okay. This trypsin helps in releasing attached cells with one another. So, enzymatic disaggregation is achieved with the help of trypsin enzyme, which release single cells like this, and then these enzymatically digested cells are released inoculated into the medium, and after the growth you will observe something like this.